What the parts of this picture are.
There's the nucleolus, which is the orange thing, and that is in the nucleus, which is the circle, the little circle. Then there's the chromatin, which is the squiggly lines. The yellow squiggles. Then the centrioles are in the cell membrane.
Thank you. Brilliant. Okay. So we're all good there. Life is good? Did we start looking at a picture like this?
Yes.
Excellent. And we were going to label this as DNA. And the DNA coils up around what? Histone proteins. And then we make a nice little flower shape out of the histones, and it coils into a nucleosome. And those nucleosomes coil up, knot, and curl until they make one entire chromosome.
You're not on it. You're drawing the fact that like the DNA is going to the histone. Is that like there's a strand of it and then it goes to that, or it's like that is part of it?
It's part of it. So think of it this way.
It's like a smaller...
You've worn a sweater, right?
Yeah.
You ever have a sweater that you pulled? Like you pulled a fiber of the sweater?
Yeah.
And if you just kept pulling, like the whole sweater would unravel? That's what we did here. We pulled a single fiber and we're like unraveling all of the DNA.
That is like one of the parts of the DNA? Like, is it... It's not like the adenine, guanine stuff or the...
That is.
Is it?
So all the adenine, guanine stuff is that stuff right there.
And that's what the chromosomes...
The chromosomes are made out of that adenine, guanine stuff wrapped around little protein balls.
Okay.
Yep,
That makes more sense.
that's exactly what it is. So this makes one big chromosome, which is what this whole big picture is, right? And did we label what the two individual pieces are called yet? And did we say that the sister chromatids are identical?
Yeah.
Brilliant. So this side is identical to this side. And it gets stapled together in the center by a what? Fantastic. Chromosomes are only visible during nuclear division. So I need to insert another. We're looking at this whole big thing is the cell cycle, right? The only time you're ever going to see those chromosomes is during this wedge of the cell cycle. They're never visible at any other time. So here's G1 right there. G2 is right here. S is here. These pieces all add up to make one phase that we call interphase. When you see a circle divided like this, do you naturally interpret it as a pie chart? So you are currently assuming that interphase takes a lot more time than mitosis because it represents like 85% of the pie as opposed to like the 15% of the pie that mitosis and cytokinesis occupies. That's how you look at that? Good, that's how you should look at that. That is precisely the correct assumption. Good graphical interpretation, someone taught you well in your past. Who should we blame it on? How about 6th grade math teacher? Do we like that person enough to say, yeah, they taught me how to do that? 2nd grade math teacher? Are you sure that was a skill you learned in 2nd grade? She's like, sure, absolutely. I liked her enough. Why not? Or him. And why not? Here's the series of circles I want you to sketch. This one here on the left. So you should have 1, 2, 3, 4, 5 circles total. well, 4 circles and then like 2 circles that got mushed together. I don't know if there's actually like a geometry name for that. I'm assuming that was actually abundantly more than 5 seconds, so we're good. What we're about to look at is that small, tiny wedge in which actually an awful lot of stuff happens. That small, tiny wedge on the circle is mitosis. And these are the individual steps that occur within mitosis. They always occur in the same order, and the same events, well, occur every time in each phase, because that's how you know it's that phase. The first phase is called prophase. It's this one right here. And you know that it's not a cell in interphase, because the initial cell that we had drawn up here was an interphase. So let's, pause to prophase. During prophase, the nuclear envelope dissolves. It disappears. So that's something really important. You'll notice there's no nuclear envelope there holding anything in. Spindle fibers, these like strange lines right there, they start to form. They're projections that come off of the centrioles. So they're part of what the centriole makes. Consider the centriole at this part like a fishing reel and fishing line. The centriole is the reel and the spindle fiber is the line. So it's just projecting them out, and it's going to attach to a chromosome at the centromere. That's what happened right there. This particular image that I happily just took from your textbook, this image divides prophase actually into 2 phases, prophase and prometaphase. Most textbooks lump these 2 images together as one. In prophase, the chromosomes condense. So chromatin condenses into chromosomes, so those little X's become visible. There's no longer a nucleus, and the spindle fibers attach to the centromere. All of those things happen in prophase and sometimes divided out into prometaphase.
Wait, so the chromatins turn into chromosomes,
Mm-hmm.
the nucleus disappears,
Mm-hmm.
and what else happens?
Spindle fibers form and attach to the centromere. Yeah, it's the double layer membrane that separates the nucleus from the rest of the cell. Do you think it was intimidated? I tried really hard right there. Yep, so spindle fibers come out of the centriole and attach to the centromere. We then move on to metaphase. So after all of those events have taken place, the 2 centrioles that have migrated to opposite poles or sides of the cell, it would be easier to interpret it as poles if it were going north-south. So do me a favor and just turn your head. And magically we have something that looks like Earth. We have North Pole, South Pole. Okay, that depends on the direction you turned your head. And an equator, right? So we have a game of tug-of-war going on between the centrioles at opposite poles. Both centrioles pull equally hard. And because of that, the chromosomes get aligned at the center of the cell. It's like a game of tug-of-war, where if both teams are pulling equally hard, the little ribbon at the middle of the rope is directly in the center. That's exactly what happens. The centrioles pull equally hard to make sure that they align down the middle. That middle is either called an equator or, most complicated name ever, the metaphase plate. So chromosomes align at the metaphase plate during metaphase. That's a question on your test. They specifically ask you what phase chromosomes align at the metaphase plate. I hope you all say metaphase. That would be delightful.
So, what person metaphase?
That dashed line right there, it's the region right down the middle of the cell. Now, because I've never been one who's much for following rules, tug of war to me seems like it would take too much effort, which is why I was not on faculty tug of war at Spirit Games time, because that seems like a lot of work. I think it would be much, much funnier and much more diabolical to just go out to center court and cut the rope in the middle. Why? They go flying in opposite directions, right? Diabolical, but yet entertaining. That's what happens during anaphase. The centromere that's holding the sister chromatids together gets cut, and the chromatids go flying to opposite sides of the cell. So that's anaphase, and that's what you see going on here. The centromere cuts, and chromatids are pulled to opposite sides of the cell.
What gets cut?
The centromere. So I think yesterday we described the centromere as like the staple that holds two identical sister chromatids together. If I pull the staple out, they're no longer held together.
So is the metaphase plate not an actual like line? It's just like an imaginary.
It's like a region, a general area. Sort of like the equator isn't like actually drawn on Earth, but we know where it is. Well, I've never actually been to the equator. Somebody might have drawn it, but I doubt it. Seems unlikely. Yeah. You ever passed the 45th parallel, though? They put up a sign for that. You guys know what I'm talking about? Yep. That's how you officially know you've made it up north. Head north on I-75, and you pass the line that says, you've just crossed the 45th parallel. Like it means something to anyone when they pass it. I may or may not have stopped there once to take a picture. Because the spindle fibers are pulling equally hard on the centrioles, the chromosomes align at the metaphase plate. Once this migration of these chromosomes has happened, they move all the way over to the side of the cell, and you'll notice in the last phase of mitosis, we have like this strange purple region forming again here on either side of the cell. That purple region, hang on, every time today when I have used this without warning students, they get scared. I am about to use the laser pointer that makes a noise. Please do not be alarmed. Every class I've done that and not warned them, they've been like, it's really the scariest thing you've heard today. That region is the newly growing nuclear envelope. So we're having the establishment in telophase of two new nuclei. And you'll notice what happened to my pretty little lines or X's that used to be my chromosomes and sister chromatids. Are there lines or X's here anymore? It's like a knotted ball of yarn, much like it was in prophase. So chromosomes unravel. They unravel, a new nucleus forms. Some people get very confused by this next part because it occurs simultaneously to telophase. You see these two indents right here, one on the bottom and one on the top? That is not part of telophase. That occurs at the same time as telophase. It's not telophase. That part is part of cytokinesis. Cytokinesis, C-Y-T-O-K-I-N-E-S-I-S. Cytokinesis is the division of the cytoplasm. It's the division of the cytoplasm. And it happens in this animal cell by the formation of what we call two furrows. So do me a favor and furrow your brow. You know furrow means indent, right? You get that lovely indent right up there? So when you hear the word furrow, I used to try and use like farming furrows because, you know, farmers make furrows to plant, but then it turns out most students don't know anything about farming. So apparently Apparently you know a lot about furrowed brows, but nothing about farming. A furrow is just a fancy term for an indent. So this furrow forms in order to cause the cell membrane here and here. They get closer and closer and closer together until they completely touch, making 2 new daughter cells. So the furrow forms in cytokinesis to make 2 new daughter cells. Nope. It occurs simultaneously too, but is not part of mitosis. Specifically, sometimes you will see this referred to as the cleavage furrow because a meat cleaver, its intention is to cut something, right? To cleave means to cut. So the cleavage furrow's intention is to cut this into 2 new cells. This does not happen in plants. Plants can't do that. Why can't plants make a furrow? Cell wall prevents it. So instead, in a plant, directly down the center of the cell, a cell plate forms instead. And it's a cell plate made of cellulose. 3rd time today I've gotten hiccups. Tried holding my breath first hour and then I almost passed out. And they were like, please don't do that. So these pictures down here are exactly what you just looked at, but from a different textbook and a different color. They're not why I show you this image. What you've seen is the cartoon version of mitosis. What's more interesting is what it looks like in real animal cells. So this is what mitosis appears like in real animal cells.